Hello everyone, this is Katish here. In this video, I'm going to go through the AQA Biology, Paper 1 Higher Tier from 2020. This is the last question in this paper. This question number 7 is on the topic of infection and response. This is mainly testing the content on the triple science content or the separate science biology content. So if you're a combined science student, this doesn't apply to you. Monoclonal antibodies, MABs, are usually made using mouse lymphocytes. Candida albicans infection produces serious symptoms in patients with a poor immune system. Recently, scientists have produced MABs to Candida albicans using human lymphocytes produced naturally after an infection. Candida albicans lives in the throat of infected patients. A sample is taken from the throat of a patient with a suspected Candida albicans infection. The sample is transferred onto a microscope slide. Describe how the MABs and a fluorescent dye could be used to see any Candida albicans pathogens on the slide. Now, this is particularly testing students' knowledge about growing microorganisms. Again, which is tested only for separate science students. So going back, they're saying that these antibodies, MABs, are usually made, right? So we've got the monoclonal antibody, let's say that's the monoclonal antibody. And they are saying Candida albicans is an infection. Now, and the third sentence says the MABs to Candida albicans using human lymphocytes, meaning that if that's the bacteria with these special proteins that it has on the surface, these antibodies, so if I draw the antibody in a different color, these antibodies will attach to the specific pathogen. Now, using this monoclonal antibody, we should be able to get these monoclonal antibodies to attach to this Candida albicans. So what the question is asking is, describe how the MABs and a fluorescent dye could be used to see any Candida albicans pathogens on the slide. So if that's a monoclonal antibody, we won't be able to see the monoclonal antibody. So what we want to do is actually attach this fluorescent dye to the antibody. Once the antibody and the monoclonal is attached to this fluorescent dye, and when we mix it, the antibody will attach itself to the bacteria carrying this dye, and once we wash off the excess, we should be able to see these bacteria because the monoclonal antibodies have attached to this specific bacteria. So if this is basically my slide, and let's say there's full of bacteria, it can have different bacteria. So wherever there's this bacteria, the monoclonal antibody has attached itself, and you will be able to see the fluorescent dye in this area, which will indicate that that person has this infection. So how do we write this? Bind or attach the fluorescent dye to MABs, monoclonal antibody, right? Then put the bound fluorescent MABs on the slide and rinse off. So you want the excess dye to actually to wash away. So the best way is actually to have the monoclonal antibody with the bound dye in a solution here, and then basically dip the slide inside it and take it off, and then rinse off with water after you dip it inside. So you can leave it there for about 30 seconds, take it out, and then wash it off with distilled water. The MABS will bind to Candida albicans and show under the microscope. Now it's a fluorescent dye. It might actually light up under a UV lamp as well. So, or show up or light up under a UV lamp. In the laboratory, the human lymphocyte MABS were injected into animals infected with Candida albicans. The MABS caused increased phagocytosis of the Candida albicans pathogens. Doctors intend to start a trial to give the MABS to patients severely ill with Candida albicans. Explain how increased phagocytosis of the Candida albicans pathogens will help the patients. Now, phagocytosis are carried out by a group of white blood cells called phagocytes. Now, there are different types of white blood cells. Phagocytes are one of them. Memory cells are another. So these white blood cells don't actually have a shape, and they have what is called a lobed nucleus. So what I'm drawing now is actually a lobed nucleus. They don't have a specific shape nucleus, and these cells can actually change shape continuously. And when it comes to the bacteria, this orange dot, let's say, is the bacteria, these white blood cells will simply engulf them by trapping them and basically digesting it using enzymes. So explain how increased phagocytosis of the Candida albicans pathogen will help the patients. Well, more of the, rather than writing Candida albicans, I'm going to write pathogens. More of the pathogens will be engulfed and killed by phagocytes. This, scroll up, reduces the damage to cells, tissues, or organs. 7.3. It has been shown that this MABS treatment is effective in the laboratory using both infected tissue culture cells and infected live animals. The MABS treatment for Candida albicans is now ready for clinical trials on people. Okay. Describe how the clinical trials should be carried out. Now, when clinical trials are carried out, or when we are testing for drugs, the first thing that we would want to do is actually test the drug on human tissue. And it doesn't, these tissue culture cells doesn't necessarily have to be diseased. They are usually healthy. What you're trying to do is to see if they will kill the cells, right? We don't want them to kill the cells. Then we actually test it with live tissue culture which are infected with the bacteria. That way we can see if it actually works, if the drug actually works on human tissue culture. Then we actually test it on animals like mice. I don't think they longer test on larger mammals, but we don't know. And then once we decide, okay, it's actually works on animals as well, we move on to humans. Now, human trials actually start off by first testing it on healthy humans to see if it has any side effects. We test it on first healthy volunteers, and then we test it on patients who have the disease on a small number of people, and then we test it again on a much larger number of people, and we test it using what is called a blind trial, and we make sure that there's a whole range of things that is also controlled during this trial. So how do we write an answer for the 6-mark question? Here's a model answer. The drugs are given first to healthy volunteers at very low dose to test if it is safe, or to test for toxicity and to check for any side effects. So there are 3 main things that are being tested on the healthy volunteers, right? To see if it's safe, toxicity levels, and side effects on healthy volunteers. Then it's given to patients with the disease, right? Why are we doing that? It is to test for optimum dosage, right? At which dose will it actually cure? Again, we are testing for side effects as well, efficacy, how well it's working, right? All of this is done in a double-blind trial where neither the patients nor the doctors know who has the MABS and who has a placebo. Now, the drug trials are run to large groups of people and over a long duration as well. So, they usually don't rush it. Drug testing usually takes about, for it to come to market, it usually takes about nearly 10 years. So that's a long time for a drug to be developed. Now, variables such as age, gender, health, diet, medication, other health issues, and race are carefully monitored as well. Now, this question actually links to one of the questions that we did. There was another question in this paper about, yeah, it actually links to this 6-mark question as well when we talk about carrying out a survey or testing on people. Now, going back to the last question. Now, this 6-mark question again, the answer for the 6-mark question feels like as it's much shorter than the answer that is required for the 4-mark question on the previous question. Anyway, that's the end of this video and the end of this question paper as well. I will see you in the next exam paper. Thanks for watching.